Bismillah ar-Rahman ar-Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. So we talked about the hypersensitivity, okay, and in a series of video lecture we talked about hypersensitivity introduction, type 1 hypersensitivity, type 2 hypersensitivity, and type 3 also. Now this is another video lecture which is also regarded with the hypersensitivity, and in this video lecture we are going to talk about type 4 hypersensitivity. So let me write type 4 hypersensitivity. Okay, this type 4 hypersensitivity, if we talked about the type 4, so type 4 hypersensitivity is delay or slow cell mediated hypersensitivity. So let me write in a short form, means delay. Let me write delay, or we can say slow type of hypersensitivity. So it is what it is, the delay slow type of the hypersensitivity. Now why? Because it takes almost 24 to 48 hours when their sign and symptoms are appear. So their sign and symptoms means type 4 hypersensitivity sign and symptoms may be appear after the 48 or 72, sometimes 72 hours. Okay, so after that hours, the rashes or the blisters become appear, which is the type 4 hypersensitivity, which show the type 4 hypersensitivity sign and symptoms. Now this hypersensitivity, if we talked about type 4 hypersensitivity, so type 4 and type 3. Type 4 is, it is opposite to type 1. If you guys remember, in type 1 hypersensitivity we talked that it is take few minutes to appear whenever we are exposed to some allergen. So it take a few minutes. So after some few minutes, type 1 hypersensitivity will be activated, means cytotoxic reaction may be activated inside our body, if you remember in type 1 hypersensitivity. But in this hypersensitivity it take 24 to 48 or sometimes 72 hours to appear the type 4 hypersensitivity sign and symptoms. Okay, this type 3 hypersensitivity, it involves different kinds of the cells. In type 4 in type 3 hypersensitivity, if you remember, we talked that it involves antigen and antibody. Okay, it involves what? Antigen and antibody. Once the antigen and antibody complex are formed, so then they will settle on the tissue surface, and then they will cause the activation of a complement system. Okay, and sometime what actually happen, let me tell you a very important thing. Sometime what actually happen, this antibody have the ability to kill the antigen or to kill the pollen. Pollen is any kind of thing that would just trigger the immunity. It may be useful, it may be harmful for us, means allergen I am talking about. Okay, so sometimes the antibody have the ability to kill the dust particles, or sometimes they will bring upon the other cells to kill the antigen. Okay, so that's very important. Now this type 4 hypersensitivity involves different kinds of the cells. Okay, we'll be talking in this video lecture one by one. It involves, let me tell you a little bit. It involves helper T cells, it involves macrophages, means antigen presenting cells, it involves cytotoxic T cells, and it also involves some kinds of chemicals, interferons, tumor necrosis factors, interleukin 6, interleukin 8, interleukin 2, just like these kinds of the chemotactic factor, or we can also call it cytokines. Now let's explain that what actually happened in type 4 hypersensitivity. Let's suppose the human body is exposed to an allergen. Again, this allergen is not harmful. This allergen sometimes useful for us, or let's suppose it is useful or harmful for us. So what actually happen, they will activate the type 4 hypersensitivity. Okay, so let's suppose our human body is exposed to such allergen. Now this allergen may be ingested or inhaled. So after that, what actually happen, this is what, let's suppose this is the let me draw blood vessels here, or tissue. Let's suppose this is what this is, let's suppose the skin area or the tissue area. Okay, it may be blood vessels, it may be our skin tissue, let's suppose. And these are what, let's suppose these greens representing the pollen or dust particles, animal tissue, dander, any kind of things it is that will just trigger the immunity. They will internalize inside our body. Once they internalize inside our body, then what actually happen? Here very important things comes about the type 4 hypersensitivity. This, this antigen will react with a heptene molecules that is present inside the human body. So these blue things as you are watching, let's suppose these are what? These are the heptene molecules. So let me write it. Heptene. So what actually happen? These green means these dust particles are connected with this heptene molecules. Once it is connected with the heptene molecule, so they can make heptene antigen or heptene dust complex, or we can say a complex is formed. Due to this complex, it will call upon a cells which we called it antigen presenting cell. This antigen presenting cell may be a dendritic cell, may be macrophages. So what actually happen after that? Due to this way, antigen presenting cell will be activated. So after that, what will happen? The antigen presenting cell will engulf it. So this is how the engulfation. So antigen presenting cell will engulf it. So they will be, engulfed. And after that, once it is engulfed, so then inside the antigen presenting cell, means let's suppose this is macrophage or dendritic cell. So what actually happen? A vesicle is formed. So this is what, let's suppose this is the vesicles. Inside these vesicles, these are the heptene molecules having antigen also present. Now, what actually happen after that? These vesicles will fuse with what? It is fused with, we call that lysozyme or lysosomes. It will means fuse with lysosome. All of you better know lysosomes contain hydrolytic enzyme or lysozymes. Each lysozymes will come and they will break down into small peptides molecules. So these will be converted into small peptide molecules as you are watching. So these are what? These are the small peptide molecules. After that, inside the antigen presenting cell, there are the MHC molecules are formed. Okay, so inside this means this antigen presenting cell, there are the molecules are formed which we called it MHC molecules. Usually here MHC2 molecules are formed. What MHC2 molecules are formed? So let me draw here what actually happened. MHC2 molecules are formed. So these are what? These are the MHC2 molecules. MHC2 molecules are usually present here and formed. So each, let me write MHC2 molecules. Now each MHC2 molecules will get these peptide molecules. So let's suppose they will get what? They will get each peptide molecules means each antigenic molecules. So antigenic molecules will be connected to their surface. After that, what it will happen? This means MHC2 molecules will will reach to the cell membrane area. Once they reach to the cell membrane area, then here a receptor is formed which we call it MHC2 receptor. So let me write a receptor is formed. So let me draw a receptor here. Let me change the color. Now this is what? This is the receptor. On that receptor they holding what? They holding the antigen molecules, or we can say they holding the peptides, or they can holding the heptene and peptide complex which is converting into small pieces. Due to this way, they will call upon some other cells, means helper T cells. So after that, helper T cell will also come here. So let me draw the helper T cell with this blue marker. So here the helper T cell will also come. So this is what, let's suppose this is the helper T cell. Now this helper T cell will come closer to this antigen presenting cell. Let me write here, this is what, this is the antigen presenting cell. So let's suppose here we take it as a macrophage. So this is what, this is the macrophage or dendritic cell. Once they come closer, so they have also, you know, their antigen. Sorry, they have also what? They have also their receptors. So these are what? These are the receptor of this helper T cell. Helper T cell 2. Keep it in your mind. In this type 4 hypersensitivity, they involve helper T cell 1. Okay, so this type 4 hypersensitivity involves helper T cell 1. So this is what? This is the helper T cell 1. So let me write it. Helper T cell 1. This helper T cell 1 will recognize what? They will recognize each of the peptide which is presented by the antigen presenting cell or dendritic cell on their receptors, on their surface, on the MHC2 molecule, we can also call that, because this is MHC2. After this, when it received the signal, means when this helper T cell 1 receive the signals, so then what actually happen? They will release different kinds of the chemicals. So here the chemicals are present, so then each of the chemical are released as you are watching. These are what? These are the chemical. Let me write the name of each of the chemical. You know, each chemical consists of interleukin 6, interleukin 8, interleukin 2, and they also you know, contain some other chemicals which we call that interferon gamma and tumor necrosis factor alpha. So these kinds of the chemicals, or we can say cytokines or chemotactic factor, are released from this helper T cell 1. Okay, after it is released, so then what actually happen? They will call upon some other cell. Sometime due to this this kinds of the chemicals, they will call upon, you know, some more helper T cell. So we have a lot of helper T cells. So let me draw here in a small circles. So these are what? These are the helper T cells. So a lot of helper T cells are bring at this area. Once a lot of helper T cells are bring at that area, and each helper T cell will release the same chemical factors. Okay, so they have, let me draw their receptor also. So they have their own receptors. So they will also, you know, bring some other T cells, and they will also bring, due to this interleukin 2 release. Once the interleukin 2 are released, so then they will call upon cytotoxic T cell, which is, you know, killer cells, which is a very dangerous cell. Not only the cytotoxic T cells we have, you know, activated, sometimes due to these interleukin 6 and 8, they will call upon some other dendritic cell, we can say, or macrophages. So what actually they will bring? They will bring, you know, cytotoxic T cell. So let me draw the cytotoxic T cell here. So they will bring the cytotoxic T cell. These are what? These are the cytotoxic T cells having nucleus. Now let me draw cytotoxic T cell. Okay, so this is the cytotoxic T cell, and moreover they will bring what? They will also bring the we can say macrophages. So let me draw the macrophages here. Blue, black. So let me draw the another structure here. So they will also bring, you know, dendritic cell here. So this is what? This is our dendritic cell. So the dendritic cells, a lot of dendritic cells, a lot of cytotoxic T cells are activated and reach to this affected area, where the antigen presenting cell are presenting the antigen to this helper T cell. Okay, after that what actually happen? Sometimes this dendritic cell will engulf this infected cell. Now why we call that it is infected? Because it engulf this kinds of antigen. It is not actually the antigen, it is not actually the harmful or useful substance, but it is just a substance that will just trigger the immunity. Okay. Now due to this way, this cell is infected. Due to this infected cells, sometime what actually happen, this macrophages will come and they will engulf it. Sometime this process happen. Sometime what actually happens, sometime this cytotoxic cells will release two kinds of enzyme which we call that perfumins, or they have another enzyme which we call it granzymes. Now these two chemicals or these two molecules or enzymes, what actually they do, they will cause the destruction of these cells. They will cause, they will create a pores. So a pores will be created. So a pores will be what? A pores will be created. Due to this pores, these cells will be die. So we call that it is cytotoxic. Here the toxic substance or the toxic material are the perfurins and granzymes for this antigen presenting cell. So this antigen presenting cell will be finished. So a lot of antigen presenting cell we have, a lot of antigen presenting cell can perform the same function as I explained, and a lot of helper T cells are activated, and because it is hypersensitive, so that's why a lot of cells are activated, and a lot of helper T cells will recognize each of the antigen each of the antigenic peptides. They will secrete a lot of chemicals, and some other lot of helper T cells are activated, and some other lot of these kinds of cytotoxic T cells are activated, dendritic cells are activated. So hypersensitivities occur, but it will take 24, 72 hours or sometime 48 hours. These kinds of times they need, such times they need. After that, what actually happen? After that, once each process are done, so then finally what we have a blister formation, as I am drawing. These are what? These are the blister. Blister is formed. So after that, the blister is formed. Each blister is fluid, painful, and very irritating. Okay, and become itchy as well. Okay, one thing that I forget to mention, what actually happened, this helper T cell 1 have another receptor which we call that CD8. So they have another receptor. Let me draw with the red marker. They have another receptors, as I am drawing. This is what? This is CD8. What does CD8 stand for? Cluster destinations. This cluster destination or CD8 receptors will, this receptor and this one, both receptor can recognize this MHC2 receptor having the antigenic peptides, and then the rest of the jobs or the rest of the functions or the rest of the functions are activated. Okay, what actually happened normally this process is not activated. Normally, let's suppose what actually happened, antigen means we can say that bacteria get internalized. Due to this bacteria, some of the antigen presenting cell will activate it, and they will engulf, or we can say macrophages are activated. They will engulf and they will break down it into small peptides due to this lysozyme, due to this lysosomes having the hydrolytic enzyme, and then each means and antigenic peptides are shift to this MHC2 molecule. MHC2 molecule then present it on the surface of the antigen presenting cell. That's why we call it antigen presenting cell, because they can presenting the antigen. This antigen will then presenting to helper T cell. Helper T cell will secrete interleukin 6, 8, 2, interferon gamma, tumor necrosis factors, and then they will bring upon some more helper T cell, more cytotoxic T cell, and more dendritic cells. Each dendritic cell, what they will do, they will sometimes engulf. Some of the antigen presenting cell are engulfed, and some of the antigen presenting cell are destroyed by this cytotoxic T cell, because they contain some kinds of molecules, some kinds of the enzyme which we call it perfumins or granzymes. These perfumins or granzymes can cause a destruction, means they can create the holes, and after that blisters formation happens. Okay, so this is normally happen. Normally we have not more cells are activated. Normally this is this means this systems will be activated due to the pathogen. So this is normal process. But if it is activated against the allergen, which is not a pathogen, which is not useful substance, which is not harmful substance as well, but it is just our immunity will just detect it as a potent pathogen. So that's dangerous process. That's all then dangerous process will activate it. Okay, so here one thing that's very important, you guys should have to note it, and the main point is that this type 4 hypersensitivity will be activated when the heptene molecules is present. Heptene molecules is present, these these allergen will react with these heptene molecules, then the rest of the things or the rest of the cells will be activated. If this heptene molecule is not present, so then the rest of the means the cells will be not activated. That's very important point. We have different kinds of example of heptene molecules. So let me write it. We have means cosmetic plants. So let me write. We have different kinds of heptenes. We call it cosmetic plants. In cosmetic plants, we have what? We have poison oak and poison ivy. So let me write poison oak and poison ivy. Each plants contain heptenes. Heptenes, sometimes what actually happen? Heptenes will reacted with the carrier proteins molecule, and this carrier proteins molecule will carry it to this antigen presenter. Sometimes this process will happen. So the best example of the type 4 hypersensitivity, that if we want to check type 4 hypersensitivity, so the best example of the type 4 hypersensitivity is the skin tuberculosis test. So let me write tuberculosis of skin test. This kinds of this kinds of test we should usually perform. What actually happen during this test? Let's suppose this is the hand of the patient. Let's suppose this is the hand of the patient. So what actually happen? Usually inside this hand we will inject the tuberculin. Tuberculin is actually the liquid molecular proteins which we obtain from the Mycobacterium tuberculosis. From this bacteria we can obtain tuberculin molecules or tuberculin proteins. Once we inject it, so let's suppose, okay, so here we inject what? We inject tuberculin proteins. So let me write, this is what they contain, tuberculin proteins. So let me write tuberculin. Try to remove the spelling mistake by yourself. So this tuberculin proteins will activate. What actually happen? When after 24 hours or after 48 hours, if the blister is formed, if the blister is formed, so it is what? It is the type 4 hypersensitivity. Okay, if the blister is formed on the injected site, where we inject, on that side, if the rashes appear, means the blisters are formed, let's suppose the blisters are formed, so we can predict that such patients have tuberculosis. What? They have tuberculosis bacteria, or they have this is the positive result. Okay, so that's all about the type 4 hypersensitivity. And again, such type of hypersensitivity, it doesn't mean that if this hypersensitivity is involved, let's suppose in my body, so it doesn't mean that it is involved in some other body as well. No, some of the patients, means let's suppose in 100 of the individual, so 10 individuals will activate such type 4 hypersensitivity. I hope you understand about the type 4 hypersensitivity. Inshallah, in the next video lecture, we are going to cover this type 5 hypersensitivity. Okay, so please stay tuned and continue to watch the video. Thank you so much.